We want to spend a little bit of time talking about the prediction of protein structures, okay? Because now we're very advanced in bioinformatics or information technology. We can use what we have learned in biochemistry and then use computers to help us to predict what protein structures will be like, okay? Now, you might be asking, like, why would we want to do that? But the answer to that should be pretty straightforward, because if you think about it, if you have to make a crystal structure or get the structure of every protein that you want to study, that's going to be challenging, because getting a crystal for a particular protein may or may not be easy. Okay, so if you talk to the professors that do X-ray crystallography or protein structures, they're going to share with you some crazy experiences where they could spend years trying to crystallize one protein. Okay, so yeah, that gives you something to think about, for all the undergrads out there. When you're deciding a graduate study, you have to really think about the amount of time that it's going to go into a particular discipline. Okay, now, we don't want to always have to make crystals and then get the structures every time. Okay, especially if, for example, I'm studying a protein, okay, that has a very similar brother that has already been solved in structure. Then wouldn't it be great if I can just use the brother structure and then I predict what my protein will look like? So that's the goal, what a lot of the bioinformatics people are trying to get to, to improve our ability to predict protein, okay? Structure prediction, really this is all talking about tertiary structure. Secondary structures are very easy to predict, and the reason for that is because we know certain amino acids prefer to have alpha helix, beta sheet, or turns. So that we kind of know, and from our existing database, our existing structures, we know what kind of sequences usually like to be in alpha helix or beta sheet. Okay, now the difficulty is actually in the tertiary structure. In prediction of tertiary structures, we make algorithms or computer algorithms that seek to have a solution for the global energy minimum. So that's sort of how we go about it. And global energy minimum, remember on Tuesday we talked a lot about protein folding and how you fold like the helix, and once you fold the alpha helix and the beta sheet, you don't go back to random coil, right? We talked about that. Okay, so it's pretty much the same concept here, right? So when we do protein folding or protein folding predictions, we try to minimize the energy for this particular sequence of amino acids that make up your protein, okay? So this calculation of the minimizing free energy is based on quantum mechanics approaches and assumptions about solving interactions. So knowing that, for example, we know the general behavior of what kind of hydrogen bonds will form, formation of hydrogen bonds is good. The distance between different atoms, we can calculate that as well. So we have some general principles in calculation of free energy. And as the program, and the program here is referring to your calculation, as the program looks for global free energy minimum, its search method must be smart. So that's a key. Why? Okay, because otherwise it will waste a lot of time looking at the vast possible conformation that a protein can adopt. Okay, like we saw on Tuesday, that funnel theory thing, right? If you have proteins in the random coil conformation, it could like rotate, and you have so many different possible conformations that your computer simply cannot possibly assay every different structure, and you look for which one is more stable. Okay, so your computer algorithm has to have a smarter way in deciding how to generate your structure. Okay, and then turns out The computer that scientists have developed, and one of the most popular one is called Rosetta, okay? It's an algorithm designed to predict protein structure. So turns out the way that people write these computer softwares pretty much mimics how the real proteins are folded. What does that mean? Okay. Remember we say that protein folding always begin with the secondary structure, right? We said that on Tuesday, right? How you first generate the alpha helix and the beta sheets. Okay. So same thing here, right? So in the computer programs, fragments of the protein sequence are organized into local short structural motifs. Okay. And essentially, local short structural motifs, basically, the first thing is you identify the alpha helix and the beta sheets, okay, the alphas and the betas. Okay, so you first take a big protein and then you look for segments of that protein that will fall into the alphas and betas. And then this computation is done by comparing sequences to a structural library. How do you know that you have fragments that will be alphas and betas? How do you know that? Okay, we know that based on our experience. As the entire scientific community grows, we have a lot of structures being solved. So if I solve a structure, right, I know that that means that this amino acid sequence give you this structure, okay? And I know exactly the fragments of that amino acid will fall into what kind of structures, okay? So we have a lot of databases that will tell you, okay, so this short peptide gives you an alpha helix. So the first thing we do when we have a computational derived structure is we first look for short fragments that have similarities to what we already have in the database, okay? And that will give you your alphas and betas. Then these short fragments are then packed together in ways that will give you low energy conformation. And for now, at the second step, we usually ignore the side chain details. Okay, so we only look at charges and polarity. Okay, why do we care about those? Because that will give you your salt bridges, your hydrogen bondings, your van der Waals interaction. Right? So we don't look at, for example, here glutamate and aspartate. They both have negative charges, so we don't look at the detail, meaning like how many carbons there are. We don't look at that detail yet. We just say, okay, you got a negative charge here. Most likely it's going to interact with a positive charge on a different residue. So we just look at a general behavior first. And then looking at that general behavior, we solve for a most stable conformation. For example, I'm going to use my two pens here. Let's say I have two helices, so I know these two helices can form because from my previous database has these sequence and they arranged into alpha helix from the past, like the past structure. I have two helices, okay? So I know that these two sequences will be alpha helix. Now, in the second step, I'm going to solve for what is the lowest in energy. Okay, how do I do that? Okay, for example, on this pen, I got a positive lysine here. On this pen, I have a negative aspartate here. Okay, so lysine, aspartate. So then, as my computer is trying to generate a lowest energy state for these two alpha helices, then the computer will try to simulate how they rotate and how they can bond into a stable structure. Eventually, the computer will come to the conclusion that if you put lysine and aspartate together, you form a salt bridge, okay, and then it's stable. Okay, so that's how the computer will do it. Okay, so it's going to try to find a structure such that you minimize your free energy. And how do you minimize your free energy? Try to have as many hydrogen bonds as possible, as many salt bridges as possible, things like that. Make sense? Okay, well, cool. In the last phase of your protein prediction is where the details of the side chains are added back. Okay, and then you calculate and optimize the energy minimum. Okay, so essentially the Rosetta algorithm is you go first, you identify based on similarity of your sequence with the other sequences in the database. You look for what are the local short structural motifs. So you find maybe, this is alpha helix, that's beta sheet. Okay, so you know local motifs, and then you assemble the local motifs, do some calculation. So the real calculation is actually here. Okay? The first step is really just matching, okay? Matching your sequence with the database. And then the second step is you're doing quantum mechanics calculation to solve for a low energy state. If you type Rosetta protein prediction tool, this is a website. Okay, it's developed by David Baker at the University of Washington, I think. He's very famous, so if you work in structural biology, you probably know this guy. He's not very old yet. He's maybe in his late 40s, 50s, something like that. He developed many different ways that we can predict proteins, and then he developed computational tools that you can use to do protein prediction. So if you go to this website called RoBeta, and what you can do is you can register for an account first. Okay, it's free. You don't have to pay them or anything. Of course, they appreciate donations and things like that, but it's free. You register an account, and if you want to use their software to predict a structure for a particular sequence, then you just log in, and then you throw a sequence there, okay, just an amino acid sequence, just throw it there, and then say calculate, okay? And then they're going to give you some potential solutions, okay? They don't just give you one solution, they usually give you multiple solutions, okay? And then these solutions are the likely structures of your particular polypeptide, okay? And then you can download that polypeptide. They actually will give you a protein data bank file, okay, PDF file, and then you can use whatever software that you like to open the three-dimensional protein structure. You can open it, and then you can like rotate the protein around and take a look at whatever you're studying, okay? I think for those people that are in our undergraduate program, we do have lab classes that will teach you how to use PyMol, that kind of stuff, okay? If I'm not wrong, okay? I think we do. You know, you'll get plenty of time playing with these softwares, right? But this is a prediction tool. All right, so that was protein prediction. So now we're going to get into quaternary structure. This is probably the only thing I'll say about quaternary structures, because quaternary structures, there's really not too much to say about it, because what it is basically the association of different protein monomers, okay? So we know that the primary structure is the sequence. Secondary structures are your alpha and betas, okay, the alpha helix, beta sheet, that kind of stuff. Those are the secondary. Tertiary structure, remember, is how you organize different alpha and beta together to form a different topology, okay? We saw that CATH category method to organize different proteins into their different class and different homologies, okay? So this is like a tertiary structure, okay? So you organize a bunch of secondary structure, so you get like a tertiary structure. So this is that. Tertiary structure is sort of a defined shape of your amino acid polypeptide, okay, your amino acid sequence, given a polypeptide. Okay, so we know that DNA gives you RNA, gives you protein, right? You go from, so essentially, ATG, TAA. This defines a particular protein. This is a protein, essentially a protein monomer. Okay, now what quaternary structure is when you take protein monomer and then you combine them together, and then you form protein oligomer or polymer or just multimer, okay? That's your quaternary structure. Now, quaternary structures can be divided into two types. You can have the homotypic, meaning that it's association between identical polypeptide chains. Okay, so what that means is, for example, in E. coli, I have a gene called, I don't know, ADH. I have a gene called ADH, and this is a gene. And when I make it into a protein, it becomes ADH, right? So it's a ADH protein, but the functional protein, you actually form maybe ADH dimer. Okay, so that's like a functional protein. And homotypic means that it's the same polypeptide, okay, the same polypeptide come together, like two of them come together, and then you form a dimer. And then we usually will call that homodimer, homotetramer, homopentamer, homohexamer, whatever. Okay, so homo means the same. Okay, so right, it's the same, right? So it's the identical polypeptide chain will come together. Of course, on the other hand, you will have heterotypic, which means association between subunits of very different protein structures. We will have those. Okay, so when we get into biochemistry 2 next semester, you will see a bunch of those. And what those are, for example, you can have 3 different types of polypeptides. Okay, so 3 different genes give you 3 different polypeptides, but then in order to give you the functional protein, they will actually come together, maybe into something that looks like this. Maybe that will be a functional protein. Obviously, your protein don't look like that. Okay, it's my crazy drawing. Right? So essentially, what happens is in heterotypic proteins, you have heteromers, meaning that you have different genes. They code for subunits of a large protein complex. So multiple subunits join together to form dimer, trimer, tetramer, pentamer, hexamer, heptamer, octamer, and so on. Cool? So you just generate different associations, and then that is your quaternary structure. Now, the interactions between the folded polypeptide chains in multi-subunit proteins are of the same kinds that stabilize tertiary structure. Okay? So essentially, the reason, okay, why you form quaternary structures is going to be the same reasons why you form tertiary structures. Okay? You can have salt bridges, hydrogen bonding, van der Waals, hydrophobic effect, disulfide bonding. The same type of interaction we see in tertiary structures will be used to give you your quaternary structures. Right? The only difference is tertiary structure is still in the same protein, the same polypeptide, but quaternary structure is many different polypeptides come together and form the functional protein. These interactions provide the energy to stabilize the multi-subunit structure. And association of polypeptide chain to form specific multi-subunit structure is the quaternary level of protein organization. Okay? So that's what quaternary structure is. Now, there are different ways that we can associate the subunits of your proteins and form the quaternary structure. You can have heterologous interaction, okay? And what that means is the interacting group lie in different region of the subunit, meaning that here you have a monomer of your protein, and your protein, let's say, it has 2 domains, okay? So 2 regions of the protein. We call that A and B. And what heterologous interaction indicates is that A interacts with B. Okay, so you have B, A, and then they have an interface here that interact. Okay, this is heterologous, meaning that 2 different domains on the protein come together. So they're 2 different domains, right? So heterologous interaction. What you can also have is you can have isologous interaction, and that is the symmetrical and identical interaction between the subunits. So in a way, it's like a exact interaction. Let's say you have A and B, but then you have A and B again. So then they form a symmetrical interaction. And then this is isologous interaction. And so you can actually form a structure that you can rotate, and then they look the same. If you look at the different symmetries, the reason we are looking for these things is because usually as proteins are folded, are organized into quaternary structures, usually they will have some kind of symmetrical pattern. And this symmetrical pattern could vary. It could be assembled into point group symmetry. I'm not going to go into point group symmetries. If you guys are interested in this point group symmetry, we talk a lot about this in inorganic chemistry. So we have a couple chemistry majors out there. You guys should probably know what point groups are. So your quaternary structures could assemble into point group symmetries, or they can go into helical structures, okay? Or they can go into other weird structures that we'll see in subsequent slides, okay? Now, point group symmetry essentially is referring to symmetries that will give you rotation, and then you end up with the same conformation, okay? What does that mean? For example, C2 symmetry, okay? You have an axis of rotation, and C2 means 360 divided by n, okay? C2 here, meaning 360 divided by 2 is 180, okay? So if you have 180-degree rotation, you end up with the same structure. That's what it means, okay? So 180 rotation, this guy rotates to here. So you have a 180 rotation, you end up with the same structure. That's what C2 means. C3 essentially would be 360 divided by 3, so it's 120. All right, so here, for every 120 degrees that you rotate, you end up with the same structure. Right? So for example, this guy, you rotate 120 degrees, it becomes that. You rotate another 120 degrees, it becomes that. Okay, that will be a C3 symmetry. Okay. And the C's usually just refers to that it has only just one axis of rotation. And then you can have D symmetry, which here you see how you can have more axes of rotation. But whatever, this is not super important for you guys at this moment. If you're really interested in protein structures, we have more advanced classes that you can take to learn more about protein structures. But so this is just very brief introduction. Additionally, you can also have isologous interactions, then that will fold your proteins into very complex structures. For example, you can have cubic symmetry, or most viral particles, you can have icosahedral. Okay, icosahedral means it has 20 different faces. It looks like a ball. Okay, things like that. All right, so this is when you organize your protein into their quaternary structures. They usually assemble into symmetrical shapes. So your whatever dimers, tetramers, usually they will be symmetrical. Okay, like for example, we see here, it's somewhat symmetrical. Now, you can also have proteins that are asymmetrical. For example, these are the heterotypic protein-protein interactions, meaning that sometimes you will have subunit A, subunit B. When they interact, it's very difficult for them to have any symmetry, right? Because they're two different proteins. They have two different shapes. So you can have dimers without symmetry. Now here, on the figure on the right here, it shows you that two subunits can associate by heterologous interactions and still not yield an indefinite chain. Okay? In this case, the interaction sites A, B, C, and D are blocked from reaction with more monomers by the close fit of the surfaces. So you can have different shapes that are not symmetrical, and you can have interactions between the different domains here. A very good example is this, where you have one enzyme on top, one enzyme on bottom, or a protein rather, I shouldn't say enzyme. When they interact, obviously here you see that they're not exactly a symmetrical protein, but that is because they have two different, it's an assembly of two different polypeptides. That should make sense, right? Because for two different type of protein, it's hard for them to have a symmetrical shape. So as a result, they usually end up in asymmetrical shapes. Larger quaternary structures, we can have structural proteins, okay? Here what you see, actin filament, you can have multiple subunits, and then they will come together, and then they form a large oligomer. Okay, actually you can form oligomers. You don't stop at a particular like dimer or tetramer, but you form a very long oligomer. Okay, actin in particular is a good example. These are like helical structures. A good example is also the tobacco mosaic virus. This is if you guys take virology, you'll learn actually a lot about this virus. This is a very interesting virus. In this particular virus, protein subunits form a helical array about the helically coiled RNA. So here, this is RNA structure down in the middle, and then you got proteins that sort of surround your RNA. Okay. And obviously you can see that they can form large protein structures, which you can actually see using electron microscope. Now, viral particles Like I said, usually they're icosahedral in shape because it maximizes the amount of space you can have. Icosahedral is the closest shape that you can have to a sphere. Now, proteins usually don't end up in spheres. Right? You don't have spherical shapes. You usually have defined symmetrical shape that end up in a compartment that looks like a sphere. And in this case, it's usually icosahedral.